What we are going to be looking at in this unit when we're talking about cancer and disease is mitosis and meiosis. Mitosis and meiosis. So these are two different ways that cells divide. Mitosis is the way that cells divide in order to grow, repair, make new cells, all of that good stuff. Meiosis is the way that cells divide in order to make sex cells, which are sperm or eggs. So we are going to start off with talking about mitosis. So when we're looking at mitosis, this is the division of somatic cells. And when we say somatic cells, those are the normal cells that make up your entire body. And this creates identical cells. Because if, like, for instance, I got a little cut on my finger here, when those cells divide and repair that wound, I want them to make more skin cells. I don't want them to make like bone cells or something like that. I don't want like a little piece of bone on the outside of my body. So they make identical, and we call them diploid daughter cells. So somatic means normal body cells. So that's your liver, your heart, all of those different things in your body. Daughter cells are the cells that we get out of that mitotic division. And what diploid means is that each daughter cell has 2N, or a full set of genetic information. So whenever in like math we have a variable, it's just a placeholder for a number. For us, N is our placeholder for chromosomes. So you know that in your body, you got half of your chromosomes from mom, or you got N chromosomes from your mom. You got N chromosomes from your dad. So together, that makes 2N, or you have 2 copies of every chromosome because you got 1 from biological mom and 1 from biological dad. So 2N is a full set of chromosomes. And we call that diploid because di means 2. So here are all of the stages of mitosis. We're going to go through each one, and then I'm going to shoot you guys off into breakout groups so you can look at them more in depth. So in interphase, your normal body cells spend about 90% of their lives here. Some cells don't really divide frequently at all either. Some cells spend 25 years doing interphase before they actually divide. So like on my leg, when I was little, I had a mole removed because I thought it might be cancerous. So they had to cut really, really deep into my little 2-year-old leg, and they cut some of the nerves out. So for a very, very long time in my life, I didn't have feeling on a little area that was probably about that big on my thigh. This was kind of fun because, you know, being the only girl with 2 brothers, we would try to do stuff to gross each other out. So I would like poke myself in that area with a needle because the nerve cells never divided and repair themselves. So I couldn't feel it until one day I could feel it. And I was in my mid-20s, and those nerves finally grew back after like, you know, over 20 years of not being there. So in interphase, what happens to these cells is 3 subphases. The first one is G1. This is where your cells are going to grow. Cells are getting bigger. They're just hanging out, doing their cell things with their cell friends. And then they get to a certain point where they're almost too big. They're almost too big. And they think to themselves, well, you know what? I should probably just divide into 2 cells because I'm getting too big. So then what happens is a signal gets sent to the cell for synthesis. Synthesis means to copy. So they're making a copy of all of their DNA because when that cell divides, each cell needs to have all of the DNA from that whole cell. The G2 phase is they grow some more and they physically prepare themselves to divide. So they got to get in their mental headspace, got to get in the game to actually divide. Next, we have prophase. So in prophase, the chromosomes condense. So if we think about like DNA, big, long, stringy strand molecule. When we say the chromosomes condense, what we mean, and actually I have a little Mardi Gras beads right here. So what happens is the chromosomes will actually wind and wind around each other, and then That little tight knot is what forms the X-shaped chromosome that we think of when we think of chromosomes. So that's called condensing. They're going from a single strand of DNA into that nice, neat little X shape. The other thing that happens, and we can see it in this picture, is spindle fibers form. So if you look at those little orange squares with like the little blue whiskers that come off of it, those are spindle fibers. I like to think of those little orange guys as like a little guy in a fishing boat, and each one of those blue lines is a fishing line. And their goal is to try to catch a chromosome. Okay, their goal is to try to catch a chromosome. So in prophase, we can start to actually physically see the chromosome. In metaphase, what happens is the chromosomes are going to line up right down the middle in a single file line. And on each side, we have those little orange things. Those are called centrioles, the little fishermen in the boat that I said before. And what they're going to do is they're going to go and try to catch the chromosome with their fishing line. So the spindle fibers are like the fishing line, and they're each trying to catch half of a chromosome. If a chromosome doesn't quite make it to the middle, then we can get some mistakes that happen where you can get too many chromosomes in your new cell, not enough chromosomes, lots of stuff can happen. So in a cancerous cell, when something like that would happen and we'd get a mistake, the cell wouldn't care. It would just keep dividing even though there was a mistake. In our normal body, what would happen is that cell would die and it would undergo a process called apoptosis, because we don't want a cell that's going to malfunction by having too much DNA or not enough DNA in our bodies. So all those chromosomes are lined up in the middle. What happens next is those chromosomes get separated. So they get physically pulled apart in anaphase. Anaphase, they go away from each other. So they're reeling in the fishing rod, okay? So sometimes a mistake that can happen is a chromosome doesn't get split and the whole X goes over to one side. When this happens really, really early in cell division, when we are fertilizing an egg, this is actually what causes Down syndrome. So kind of a unique, interesting thing. The last part of mitosis is telophase. This is where we actually have two separate groups of our genetic information. Our new nucleus starts forming around each one, and we almost have two brand new cells. Lastly, what needs to happen is cytokinesis. So mitosis is the division of the DNA. Cytokinesis is the actual division of the physical cell. So you can see here that both sides of that cell are like pinching in on each other and pinching in that membrane, and they're creating a physical division between those two cells. Okay, so I am going to stop recording.